Okay, this video is the Michael Brownlee paper on diabetes. This is for doctors only. If you're a non-doctor, go ahead and listen, but I'm going to focus this conversation on doctors. Just so you guys know, diabetes being caused by high dietary fat, that's been known since at least 1927, the J. Shirley Sweeney study. This is Michael Brownlee. This guy's a genius. This is one of the best papers ever written about diabetes. It's his paper on the unifying theory of diabetes complications. He won the Banting Award in 2004, and he gave the Banting lecture. That's what the paper's all about. And what he figured out was that too much fat goes into the mitochondria, and it sends too many electron carriers to these complexes, 1, 3, and 4. In particular, it's at complex 3 where it starts to back up. The proton gradient goes too high within the intramembranous space, and then electron transport on the intermitochondrial membrane goes backwards, and it starts dropping electrons down onto oxygen in the mitochondrial matrix, which forms superoxides, a free radical with an unpaired electron in its outer orbital. Under normal conditions, a small amount of superoxides are formed, but they can be neutralized by superoxide dismutase, SOD. The Mn is for manganese, which is characteristic. Okay, but superoxide dismutase can neutralize the superoxides. That's important. However, when there's an excessive amount of superoxides, they overwhelm SOD, and you will start to produce additional free radicals within the inner mitochondria space there in the matrix, and that will damage mitochondrial DNA. I'm not going to get into all the details, but it'll also lead to damage of DNA in the nucleus, and that'll activate nucleus repair enzymes. I'll get to all that in a moment. I'm going to actually show you the paper. You can get this paper. You just go to diabetesjournals.org. We're going to go through the paper page by page. Okay, so here's the first page, and the key point here is that he says he had type 1 diabetes, and he would have otherwise died if they didn't have insulin available when he was a young child. So he devoted his whole life to studying diabetes, this Michael Brownlee guy, and the guy really is a genius. It's pretty amazing. So he had noticed from reading all the journals that there were 4 major things causing diabetes complications. Called them, I made a mnemonic here, PHAD, Polyol pathway, hexosamine pathway, AGEs for advanced glycation end products, and then D for diacylglycerol, which activates protein kinase C. And usually they're going to talk about protein kinase C, but just remember this, it's like FAD, PHAD, and it becomes real easy to remember. And the first 2 aren't even that important for our purposes, just the A and the D right there. I've been told by really smart senior internal medicine doctors this would be the most important paper that a doctor could read to start getting a clue about the significance of the role of nutrition in health. Okay, so the first thing he talks about is the polyol pathway. The polyol pathway means that normally glucose just runs through glycolysis, but when there's excessive amounts of glucose in the cell, so intracellular hyperglycemia within the cell, hyper amounts of glucose in the cell, the aldose reductase enzyme will convert it to sorbitol. Okay, but the important point is normally that aldose reductase enzyme is supposed to be processing toxic aldehydes to inactivate them. This reaction has a cofactor of NADPH. So when you run this reaction from too much glucose in the cell, you'll deplete NADPH. NADPH is also a cofactor for keeping glutathione reduced, which you need because it's a very important antioxidant. So the bottom line is when you run glucose through this pathway, you use up your NADPH and you no longer can maintain glutathione reduced. So you then are more vulnerable to oxidative stress. So that's the key part of that for our purposes. Okay, the next thing is you can run the hexosamine pathway. I don't have the picture right here, but that's not that important for our purposes. Just know it exists. Okay, now here is the next thing called advanced glycation end products, AGEs. And you're going to form those off of glycerol 3-phosphate. And the AGEs, especially methylglyoxal, is the main sort of precursor to all this. You can then glycosylate or glycate proteins inside the cell, in the extracellular matrix, and also in the blood, like albumin. Those can be taken up by other cells on what are called RAGE receptors, receptors for advanced glycation end products, and that leads to increased amounts of reactive oxygen species production, ROS, activation of nuclear factor, kappa B, And then just more inflammation. Inflammation's always, prothrombotic. It's bad. Okay. The 3-phosphoglyceraldehyde, the intermediate in the middle of the glycolysis cycle, because its enzyme is inhibited, glyceraldehyde 3-phosphate dehydrogenase, it will start to run side pathways. In particular, it gets converted to DAG, diacylglycerol. The diacylglycerol then gets converted to protein kinase C, and that just unleashes a whole bunch of really destructive pathways. In particular, it does the opposite of good health. Good health is to run endothelial nitric oxide synthase and make more nitric oxide, vasodilator, and inhibitor of platelet aggregation. With diabetes type 2, you do the opposite. You'll diminish nitric oxide, you'll increase endothelin, which does the opposite. It causes more clotting in the platelets, okay, and it's a vasoconstrictor. So that's double bad. You'll also block the plasminogen activator inhibitor. So basically you can't do fibrinolysis. You're more prone to forming blood clots. You'll activate nuclear factor kappa B, so you're more prone to inflammation. You'll also activate NADPH oxidase, which means you're going to make more superoxides. You're going to have more oxidative stress. Okay, so the bottom line is all these bad pathways are unleashed. And then the question was, well, how does one explain how they're all activated? This just shows the hexosamines pathway, which we don't really care about too much. Okay, so the way that these all happen is because what we just talked about a moment ago. When you have excessive amounts of fat getting into a cell, and it's especially excessive amounts of saturated fat, but eventually all types of fat will lead to insulin resistance. Not as quickly as sat fat, but they're all going to lead to it. Sat fat, you produce additional electron carriers because you have to do more beta oxidation to run it through Krebs cycle. So anyways, so things back up at the level of complex 3, then coenzyme Q is overwhelmed with too many electrons. It starts to drop them down into the matrix, and then they get picked up by oxygen and it becomes superoxide, O2 minus, with an unpaired electron in its outer orbital. And again, under normal conditions, it's neutralized by superoxide dismutase, SOD, to become eventually water and oxygen, harmless. But if there's too many of these electrons forming too many superoxides, you'll then start to damage things with free radical reactions. Now, you can prevent all of this with UCPs, uncoupling proteins, which really are weak acids. And they'll just pick up a protein and carry it from the intramembranous space over here down into the mitochondrial matrix. Okay, then they become deprotonated, diffuse back across, grab a protein, and come back. So that's what an uncoupler does. They also found if they added excessive amounts of superoxide dismutase to the mitochondrial matrix, they could prevent the downstream complications from happening. So they could prevent this. That's how they knew that this is what does it, because they could block it with uncoupling proteins, and they could also block it by adding extra superoxide dismutase. And so Brownlee and his group did all this work. They checked and double-checked everything in multiple different ways. Okay, so here's a summary of what happens. The thing you have to know that's kind of bizarre is the glyceraldehyde 3-phosphate dehydrogenase enzyme of glycolysis. It actually is also involved in DNA repair, and it shuttles back and forth between the cytoplasm and the nucleus of the cell. And that's how it's able to travel into the cell where it encounters this other DNA repair enzyme, polyadenylyl ribosylating protein, and that will ribosylate glyceraldehyde 3-phosphate dehydrogenase and inactivate it. Once this becomes inactivated, the things proximal to that within glycolysis will start to accumulate. Glucose accumulates, and it gets run through the polyol pathway. Fructose 6-phosphate accumulates, and it gets run through the hexosamine pathway. Glyceraldehyde 3-phosphate accumulates, and it gets run through A for AGEs to make advanced glycation end products, okay, first via methyl glyoxal. And then it also, the glyceraldehyde 3-phosphate also goes under the side reaction to become diacylglycerol, which just means 2 fatty acids on a glycerol, rather than like a triacylglycerol is like a triglyceride, which would be 3 fatty acids on a glycerol. So diacylglycerol, 2 fatty acids on a glycerol molecule, and that gets made into protein kinase C, which then unleashes a whole bunch of really bad pathways, like we just talked about. So what started the whole process? The damage to the overwhelming of the intermitochondrial membrane electron carriers, leading to a backup of those carriers for their electron transport, leading to superoxides. The superoxides being transmitted to the nucleus, damaging nuclear DNA, activating the PARP enzyme. The PARP enzyme damaging glyceraldehyde 3-phosphate dehydrogenase enzyme, which then travels back to the cytoplasm, and glycolysis can't run, so all this stuff backs up, and you get all these side pathways. Okay. Now, one of the things some people will say about this paper is, well, in the paper he emphasizes that hyperglycemia inside the cell will make this happen, and that is true. So when you have hyperglycemia in the blood, the high blood glucose goes to cells that are vulnerable. The vulnerable cells are the ones that have constitutive uptake of glucose, meaning that they cannot regulate the amount of glucose they take up into the cell. They take up the amount of glucose in proportion to the amount that's in the blood. So the classic examples of that are the diabetes complication locations. So in the retina, the retinal cells and retinal endothelial cells, they cannot control their uptake of glucose. They just take it up into proportion to the blood concentration, so you get diabetic retinopathy. Then in the kidneys, the mesangial cells, for example, cannot control their uptake of glucose. They just take it up in proportion to blood concentration, so that's how you get diabetic nephropathy. In the peripheral nerve cells cannot control it, so you get diabetic neuropathy. And the endothelial cells of the arteries, they can't control it either, especially the capillary for microvascular disease. And so you get a microvasculopathy from diabetes. And it's really bad in the small arteries. That's why you get all these problems down in the toes. And then you have to have toes amputated. They also have a lot of problems in their brains. Diabetes type 2 patients are some of the stupidest patients. I'd probably say the stupidest patients are kidney failure patients. Okay, and then you get some pretty stupid patients with, obstructive sleep apnea, of course. In terms of large numbers of mildly cognitively impaired, diabetes type 2 is super common. Okay, and of course there's just patients that are stupid in general, alcoholics and IV drug addicts and all that sort of stuff. But it's very common because there's multiple reasons why the pathophysiology of diabetes impairs cognitive function. I talked about that in other talks. I think I made a talk something like, does diabetes make you stupid? Okay, but what I wanted to get at is the intracellular hyperglycemia is only relevant for those cells which cannot control their uptake of glucose. But the key thing to know is that in the skeletal muscles, they cannot control their uptake of fat, in particular saturated fat, and that causes insulin resistance because it does the same thing. It gets into the mitochondria too fast, makes too many electron carriers, too much NADH, to some extent FADH2 as well, and this leads to overwhelming the inner mitochondrial membrane electron transport chain complexes. So they start going backwards and dropping electrons down into the matrix onto oxygen to make superoxides. That's what it's all about. And I'm going to show you why that happens, because now the question you're asking yourself is, well, why does too much fat get into the mitochondria? And here's your answer. What they found was that fatty acids can get into the skeletal muscle cells in proportion to their blood concentration. They can intercalate into the outer leaflet of the plasma membrane and then flip into the inner leaflet and then pass into the cytoplasm. So this is called the fatty acid flip-flop maneuver. Okay, so it can gain a proton to facilitate intercalating into the outer layer of the plasma membrane, then flip here. And they found this out to be true because they tried inhibiting the fatty acid, the known fatty acid transporters, and even when they had all of them blocked that they knew of, it just kept coming in the same. So it wasn't to do with the fatty acid transporters. And so this is why fat gets into the skeletal muscle faster than glucose does postprandial, because it can just flip-flop across this membrane. Glucose is slower. It's dependent on the whole process of insulin being made, insulin binding to the insulin receptor, insulin interacting with the cytoplasm to cause the glucose type 4 transporters to travel up from the cytoplasm to the plasma membrane. So anyways, this paper by Michael Brownlee, again, it's called The Banting Lecture 2004, The Pathobiology of Diabetic Complications, A Unifying Mechanism by Michael Brownlee. It's available at the website on the internet, diabetesjournals.org. You can try going to PubMed. You might not be able to get to the paper. Now, one thing I know you're going to tell me, oh, you don't have time to read the paper. And I'll say to that, BS. All you got to do is print out the paper and then put a little table in your bathroom. Every time you go in there, read that paper. You will read that paper so fast it's not even funny. I read the paper in one day, okay, in one day, just from going into the bathroom. It's easy to do if you just do what I say. Print the paper and have a little table in your bathroom. Everybody goes into their bathroom. You'll be amazed how much you read if you start reading papers in your bathroom. So anyways, and once you learn this, you'll be like, holy shit. And diabetes is type 2 is easily curable. The patients just lose weight. Look at the Roy Taylor. He wrote an entire book on that and a bunch of papers on that. All his type 2 diabetes, less than 4 years duration, who lost about 30 pounds of body weight, reversed their fatty liver, and cured their type 2 diabetes. Walter Kempner, back in the 1940s, 50s, and 60s, and 70s, he routinely cured his type 2 diabetics. They would just lose weight, and they'd get healed from type 2 diabetes. Nathan Pritikin had the same experience, as did John McDougall, etc. So there it is. There's your education about type 2 diabetes.